All right, before you go into the field, you're going to need to have some materials to collect plants. So everything I have for me in the field here is in this bag. So you need a bag or a fanny pack to take with you in the field. Nice to have lots of pockets. The first thing you'll need, which I keep in this inner bag here, are some plastic bags to collect plants in. It's helpful to have some different sizes. I usually keep some smaller bags like this and then some larger kind of kitchen-sized plastic bags, kitchen-sized trash can plastic bags to collect with. So I keep lots of bags in the back here. In this outer pocket here, I keep a variety of items. The first thing I have is a GPS receiver to take latitude and longitude of the collection. These days you can also get this information on your phone, so you either need a phone or a GPS receiver. You'll need a collection notebook. These from forestry suppliers are great. They're right in the rain, which means when they get wet, your pencil, what you mark in pencil won't come off as easy. But in this collection notebook, you're going to note location and habitat, latitude and longitude, frequency, this kind of stuff for each of your collections. So you need a collection notebook like this. You'll also need a pencil to write in your collection notebook. You'll need a Sharpie to write your collection number on your collecting bags. I also keep a hand lens to look at things up close in the field to verify field identifications. I'll also often keep some extra batteries for my GPS receiver. You'll also need some things to take plant material. This is a hori, which is a Japanese digging tool, but you can use any kind of small trowel or anything to dig plants to get underground parts. And then lastly, you'll need a good set of clippers to clip off material of woody plants. These are the clippers I use in the field. These are all the things you'll need in your collecting bag. Yours will have maybe some different items, but this is the basic set of equipment you'll need to go in the field. All right, we're here today at Wichita's Chisholm Creek Park to show you how to collect plants. We already know what's in our collecting bag, what we're going to need to collect, so let's go out and grab something. Now, let's say you were just doing a survey of this site. You wanted to know what plants were on this site. So what you do is you would try to collect things that were reproductive and then go out and key them out later and eventually make a list of everything that was on this site. Now it's mid-July, so there's not a lot of things out here that are reproductive at this point, but I do see this Asteraceae here in front of me that's reproductive, so this would be something good to collect. First thing I always do whenever I get to a site is I always turn on my GPS receiver so it can be getting the coordinates for this point. Set that right there. And the next thing we want to do is we want to pull out our digging tool, because we want to get underground parts. Underground parts, if possible, for herbaceous plants are really critical for identifying plants. So it's critical to get underground parts if you can. So here I'm going to go down to the base of this. I'm going to pull some of the adjacent vegetation away so I can more easily get at it. And I'm just going to start digging around the base of this plant. Trying to get those underground parts. So now I've got a nice little soil bit here. Looks like I've managed to cut myself. So we can kind of pull the soil away from that, and look, we've got a nice underground portion of this plant. So then we want to take it and put it in a collection bag. Okay to bend the plant because you're going to be bending it later anyway. And you want to take out your field notebook, and this is where you want to write information about the site. I'll show you what this record looks like, so I'm going to write this here in a few minutes. Basically, you want to write, first of all, you want to write the location. And you start by exclusive to inclusive. So we're in the United States, we're in Kansas, we're in Sedgwick County, we're in the city of Wichita, we're in Chisholm Creek Park, and then we're going to use the coordinates we take to come up with a distance from a road or a distance from a parking lot, and that'll be our location. We also want to note habitat. So we're here in this restored mixed grass prairie. We're in full sun. You want to note something about the commonness, the frequency of the plant you're collecting. So this plant is, let's see, you look around, you can see quite a bit of it, so I would say it's frequent. That would be the term. If a plant is less common, you might say that it's uncommon or even rare. You might note that it's the only plant that you saw. It can even be more common than frequent. You can say that it's locally dominant, that kind of thing. So once we've noted all of that material, so we've noted location, frequency, habitat, we've written latitude and longitude from our receiver. So we use our receiver to write down latitude and longitude, and also elevation. So this plant for me is going to be Beck 1781. So I might even write Beck 1781 on this bag here. I just might write 1781 here, just so I know that that's the plant that I'm dealing with. Put our digging tool back. And now we're going to go over and grab a woody plant while we're here. This is nice and reproductive. So we're going to take, for woody plant, obviously we can't get underground parts, but we can take several nice cuttings of the tree there. We can put those in the same bag, and since these were from the same location basically, we can use the same kind of distances and everything, you know, Chisholm Creek Park, distance from a parking lot, that kind of thing. We want to note a different habitat though, right? We want to note that this was a forest edge, forest prairie edge, full sun again. So we'll use the same location and the same coordinates, but the habitat will be different. So we'll have a separate second record for this species. And we'll also have latitude and longitude for it again. And we might even write 1782, because that's our next number for this one. So we know we've got Beck 1781 and Beck 1782 in this bag. And that's it. In a typical day, you know, you might go out and you might collect a lot of things, and if they're all basically from the same site, you can put them all in the same bag as long as space allows. You'll have a different bag for each site. Each site again will have the same location. Yeah, that's basically it. And now we're going to go back into the lab and press these plants. Okay,
A little bit about the kind of data you want to take. This is a label. So this is the label that's going to go at the bottom right-hand corner of a specimen. And this label contains a variety of data. First of all, it tells you what state the thing is from, the county it's from, its family, the taxon. Then it lists location. So the location here is Cascade Springs, west side of Highway 71, 8 miles south of junction with Highway 18 bypass and Hot Springs. So that's the location. The habitat, epipetric, just means it's found on rocks. On both sides of west running small calcareous canyon with juniperus, pinus, vitis, prunus, and toxicodendron partial shade. So that's your habitat. Then we have our latitude and longitude and elevation. Then we have some other notes. We note that this thing was sporulating. Later we actually counted spores per sporangium, and we know that the plant was frequent. We have the collector and the collector number, the date, and some other information down here. So this is a kind of label that you want to make. And label making is something we won't go into, but basically it involves having certain information typed into an Excel spreadsheet and then exporting that information into a Word document. But you need to have this information. So we need to be able to type some stuff into an Excel spreadsheet when we're back in the lab. So we need to take some data in the field. This is a basic summary of what that data, the kind of format for what that data might look like in your field notebook. And you can use whatever kind of format you like. You can take these in whatever order you want, but this is kind of the basic information you need for every collection. Collection number, right? Your name and the collection number. The taxon that you have. Now, you don't have to know the taxon when you're in the field. You can key that out later, so you can leave that blank if you want. You can provide a basic description of that. You can say, for instance, blue solanaceae or something like that to help you kind of jog your memory of what you're dealing with. You need to note the country, the state, the county, and then the location. And the location is going to be as detailed as you can make it. You need to note habitat, and then you need to note other notes. For instance, the habit of the plant, whether it was woody or herbaceous, how common it was, that kind of stuff. And finally, you need to take latitude, longitude, and elevation. So what's my field notebook look like for the collections we made at Chisholm Creek Park? Well, it looks like this. So it's over here on the bottom right. First of all, I noted the date. I made these two collections on the 20th of July, 2021. Here's my collector number, 1781. The taxon here is blank because we haven't keyed this guy out yet. But then I go into my location. USA, Kansas, Sedgwick County, Wichita, Chisholm Creek Park. And then I note that it's 0.5 miles northeast of the junction of Oliver Avenue and 29th Street. So how did I get that? Well, another thing I took over here was the latitude and longitude, right? And you can plug that in later into Google Earth and it'll help you get locations. So let me show you what that looks like. Here's Google Earth, and I've typed in here that latitude and longitude that I took from my GPS receiver in the field. And if I hit search, it's going to zoom me down to that location. So we're zooming in on Kansas, on Wichita, and there's Chisholm Creek Park here, this green rectangle here. So a good way to take a location, to add information to a location, is from junctions of roads. So we have Oliver Avenue here, and we have 29th Street here. So we could take the distance as the crow flies from this intersection. And in Google Earth, you simply use the ruler tool. So I've set it to miles, and you click on the intersection, and then you move it up to your location. So you can see we're about 0.55, 0.54 miles from that intersection. So I'm going to round down to 0.5. So that's how I got. That's how I know we're 0.5 miles northeast of this junction between 29th Street and Oliver Avenue. Well, that's what I put into my note there, 0.5 miles northeast of Junction Oliver Avenue and 29th Street. And that's as detailed as you need to make a location. You can always add as much information as you want. However, the point is, with this latitude and longitude information, if that's on the sheet, any future worker should be able to find the exact location of that plant. The location information you provide here is just a verbal description. It's kind of a shorthand for what we have here. So then we have our habitat, restored mixed grass prairie, and it was in full sun. Then other notes, the plants were in flower and they were frequent. My 1782 collection, that woody plant, the location is the same, so I just write as 1781, so it was at the same location, but the habitat is different, right? So I note that it was a prairie forest boundary, full sun. In my other notes, I note that it was a shrub, and it was about I estimated that it was about 5 meters tall. I noted that it was fruiting, and that it was also frequent. So this is what your field notebook looks like, and notably it's in order of collection. So you can see my previous collections here that were taken. My previous collection came from Colorado, 1780, and so your book just moves forward in chronological but also in collection number order. You just simply add your collections as they go. So the point is, you can take much of this information in the field. Obviously you need to note habitat and other notes in the field, but as long as you take this this latitude and longitude information, you can reconstruct your location later on your computer. So not all of this information needs to be taken in the field. Mainly the number, habitat, and other notes and date need to be taken in the field. So that's the kind of information you would take in the field that you would type into Excel and use that to turn into a label later.
So we're back in the lab, and now we're going to press these plants that we just collected. Remember, this is your goal, a specimen like this. All specimens bear labels, which has the information that you took in the field and some other information on it. And they occupy that bottom right-hand portion of the sheet. The rest of the sheet is occupied by some stamps and barcodes and things, but basically you have the rest of the sheet to work with. So that means if you press the plant in a newspaper that's of the same size of that sheet, you'll more or less be on the right track, right? If we press our plant in a space this size, then we will end up with enough material for a herbarium sheet. And plant presses are also of this same size. So in other words, we want to have a newspaper that matches our plant press size and a sheet size. You can often find what's called tabloid style newspapers that are this size, just regularly this size, that you can find at gas stations, that kind of place. But if not, you can always cut the bottom off of a regular size newspaper to fit the exact size of your herbarium sheet. Now before we press these plants, I'm going to unload a press to show you kind of what some pressed plants look like. These are some collections I made last week. If we open up this press, here's my collection, Beck 1780, and it's of a fern I collected in Colorado, so you can see it's pressed nice and flat. Some critical things about this specimen is that I've got both surfaces, both the top and bottom surfaces of the leaves pressed, so that's important. You want to have both the top and the bottom surfaces of the leaf. You want to have the underground parts there, if present. And you want to kind of more or less spread out the material, so you don't have a bunch of material, kind of overlapping. You don't want to have a specimen, in other words, that looks like this, where everything's kind of on top of one another. You kind of want to have things spread out so the morphology is clear. Now normally when you make collections, you make duplicate specimens. So you're going to actually see that I have 3 or 4 specimens of this Beck 1780 that I collected in the field. and I press duplicate specimens. And duplicate specimens means that those specimens, one can be stored in your home herbarium, and the rest can be distributed to other herbaria. So duplicates of that exact specimen. And this is important because this allows that material to be broadly distributed so people can see and have records of that same plant at that same site in other places. So it allows people to see what plants you have here in Wichita and have them in other places. They can look at them for study, have them for records, that kind of thing. It's also important as kind of a backup, so in case something happens to the specimen here at Wichita, there are specimens in other places. Here's Beck 1779. So this is another fern that I collected in Colorado. You can see it's a small fern, so there's several individuals that I pressed here on this same sheet. Again, several duplicates of that specimen. So those are some previously collected specimens that have been dried. So now what we're going to do is we're going to press the specimens that we collected today. So this is a herbarium, this is a plant press. So the bottom of a plant press is often kind of this grid of thin wood bolted or nailed together. It allows airflow, a little bit of airflow into the press, and it also keeps it lightweight. Then the next thing you add are what are called ventilators. And ventilators are corrugated cardboard. So cardboard that has these vents that run through it. This is going to allow air to travel through this this cardboard as it dries, which will allow the plants to dry. So the first thing we do is we put down one ventilator. Then we want to put down one of our sheets of newspaper. So that first specimen we collected was Beck 1781. So we want to write Beck 1781 here on the sheet. Then we want to press our plant. So let's get Beck 1781 out of this bag. It's going to be possible to press this entire specimen on this sheet, although that's not always entirely possible. And as you can see, this one's already bent in a fairly convenient way. So we can, well, first thing we want to do is we want to make sure we get all the dirt knocked off of this underground part. You don't want to have a bunch of dirt in your specimens. So I'm going to take this over the trash can and tap that a few times. Make sure all the dirt's knocked off of the bottom of that specimen. You can also rinse them under water for a short amount of time. That'll work too. So this is already in pretty good shape. We can see we've got our underground parts there. We got a nice bend to the specimen. We are going to have to bend these down a little bit to make sure they fit on the sheet. And it's okay to put the plant in kind of an unnatural position, if you like. It doesn't have to be exactly how it would grow. In other words, it's okay to bend the plant to fit on the sheet. Everyone who examines a herbarium specimen knows that that's the case, that people have to do that. So we've got everything kind of spread out here. Not a lot of stuff is overlapping. And we can kind of, this one is ready. This one's ready to press. We can make sure these leaves, this one doesn't have a lot of leaves left on it. We can make sure these leaves are flat, sticking out there. We're going to have plenty of room for the label down here, so that's ready. So then we can just hold the plant in place, move the newspaper over, take another ventilator, and put it on top. Kind of make sure everything's set in there really well. Pressing inside is easy because there's no wind. It's really challenging to press outside in the wind because it blows your newspaper around, that kind of thing. But inside on a surface like this, it's nice and easy. So now we're going to go on to Beck 1782. They have to do some trimming on this one because remember it's that big woody plant. So obviously this is way too much material to go on this sheet. So I'm going to do a little trimming. And because plants are redundant, in other words, because they have lots of the parts are all kind of the same as you move up and down the stem, you can kind of aggressively trim. You can aggressively trim them. So I'm going to trim this one like so. I'm also going to remove some of these branches here so we can better see the stem. You can see we're endeavoring to press. You can see we have both sides of the leaves here. We have that lower shiny surface and the upper surface. I'm going to get a few of those in that position. So that's pretty good like that. We have upper and lower surface of the leaf. Things are not really overlapping with one another. So that's ready to press. And because we have more of this specimen, I'm going to make a duplicate of this specimen, so one for distributing to another herbarium. I'm also going to show you one more bit here in a second. I'm going to save this material here because we're actually going to put this in silica gel. We're going to silica dry this for molecular work later. So we're going to do that. So this came off of a single individual. I'm going to set it aside. Let's press this other duplicate. Turn this over so we get both sides of the leaves here. That's pretty good, right? We don't have a ton of overlap. We can clearly see the stem. We got both sides of the leaves. Everything is relatively spread out. We're going to hold that in place and press that. Going to put another ventilator on top of that. Then we're going to place the other end of the press on top. Make sure everything's nice and square. Get our straps. Get them kind of hand tight on each side. And we're going to really crank this down tight. And the way you do this is you put it on the floor, you step on both sides of that strap, and you crank it as hard as you can. Step on both sides of this strap and crank it as hard as you can. You'll know that the straps are tight enough when you can play them almost like a stringed instrument. They're so tight it's like you're plucking a guitar. So this press is certainly tight enough. I'm going to tuck in the straps. And this press is ready to dry. I'll show you our drying rig over here. Here you can actually see another press that's in that drying rig. This drying rig is really simple. It's a milk crate on which you set the press on its side like this, and you wrap it in a tarp, just a hardware store tarp. You kind of pinch off that to make sure it's nice and tight. Then you take a regular hardware store space heater, set it here, plug the space heater in, and turn it on. What that's going to do is that's going to put hot air below the press, and then the only way the hot air is going to be able to escape is to move in between, through the holes in that ventilator. So in other words, we're going to have hot, dry air passing through, passing in between each specimen. So I can actually feel the hot air moving up through these specimens, and so that hot air moving in between these specimens will actually dry them relatively quickly. This might take 6 or 8 hours to dry a press of this size. This press would dry much, much faster. There's many ways to dry plants. So large herbaria actually have a dedicated big heaters to dry plants. You can also simply dry plants at room temperature. If they're not super wet, they'll dry in about a week or two. You can always change the newspapers out if you think that they're really wet. There's also cabinet dryers, so herbarium cabinets that actually have forced air in them that dries plants. This is a cheap and efficient way and portable way of drying plants. It's obviously not the most, I guess, safe way to dry plants. I mean, we have a space heater hooked up to a tarp and a plant press, so there's a little bit of a fire hazard here. So you never want to leave a press. You always want to be in the room when a dryer like this is running. You never want to leave the room when it's running, just in case the thing does catch on fire. But this is an extremely cheap, portable, efficient way of drying plants that we use the heck out of here in the Beck Lab. And so once it runs like this for 6 or 8 hours, then the plants will be ready to take out of the press, just like the flat, dried press specimens that we examined here a few minutes ago. And then they'll be ready to identify. We can put them under the scope and identify them. And then they'll be ready to mount on a herbarium sheet and put in their herbarium. So that was a basic introduction on what you need to collect, how you collect plants in the field, how you press them, and how you dry them. I hope you enjoyed it. Okay, so I said I was going to show you how to preserve material in silica gel for later molecular work. So here's our bit of tissue from Beck 1782. Now what we're going to do is we're going to preserve some of that in silica gel. So the first thing we need to do is label just a regular manila coin envelope with the collection number. Then we need to put a couple leaves, pick a couple healthy leaves from there. That's all you'll ever need to do a DNA extraction. Put those in the envelope, seal it, and then put it in a bag with silica gel. Now silica gel are little beads or a little powder that absorb water, and they have this blue indicator or orange indicator gel in there to show you when it's exhausted. So you can see these indicator beads, which comprise about half of this silica gel, are either blue or orange, which means they can take on water. They're not exhausted. The blue will turn pink and the orange will turn green when it can't take any more water. So you see I have this bag here with silica gel in it. Here I have a previous sample that I've dried in silica gel, Beck 1780. If I open it up, you'll see that the tissue inside is completely dry, right? So it's been completely dried out. So now all we have to do is we have to put our sample in the silica gel, seal the bag up, leave it there for a day or so, and it'll completely dry out all of the water in that, all of the moisture in that sample. And then once it's been in that bag a couple days, then we can move it to our silica gel archive. So the silica gel archive in the Beck lab is over here. So you can keep your tissue in one of these boxes here that has a little bit of silica gel in the bottom to kind of maintain that dryness. I'll show you what the archive looks like. If you open it up, we have these baseball card boxes which contain these stacked coin envelopes. As you can see, my collection's in order. So there's Beck 1778 here. So I'm gonna put in Beck 1780 here, and then it can be stored permanently in that silica gel archive. So that tissue, all of these tissues can be used to extract DNA from and do any kind of molecular work we want to later. All right, so now let's talk about mounting plants. So your plants have come out of the dryer. Then you put them in a freezer for a period to make sure they're bug free. We don't want any bugs coming in on the plants from the environment. So you can either put them in a -80 freezer for a relatively short amount of time, like a week, or you can put them in a -20 freezer for a longer time, like several weeks, to make sure we kill all the bugs and all the bug larvae off of the specimens that might be coming in from the field or from the lab. And then what you've got is you've got a stack of plants to press, right? They're in their newspaper. They're pressed flat. They're dry. You've made labels, which we still need to talk about, at least the data. But here you can see this is a good example of what the data looks like on a label. Here we can see it's from Kansas. This plant was collected in Morris County. There's the plant family. This is the golden rain tree, Corateria paniculata. It was collected in Council Grove, 60 yards west of the Cottage Grove Hotel, so that's your location. Disturbed edge of parking lot. Here's your latitude and longitude and your elevation. This plant was fruiting. Noting the frequency, I'm saying there was 2 planted adult trees. I'm noticing the height of the trees, and also noting there was numerous escaped juveniles below. Here was my collector and number. This was a person who was with me when I collected. Here's the date, and I noted that I took silica dry material from this specimen. We've already talked about how that's done. Also noted that I took some soapberry bugs from this plant. That was from another project. Here's my duplicate info. In other words, this is a unicate. This specimen is only being deposited at the Wichita State Herbarium, or WICH. And then at the bottom it just notes, this is our herbarium. So there's your pressed flat plant with a label. This label is on acid-free paper, so it's cotton paper, so it's not going to age over time. It's not going to yellow over time. And we also mount on acid-free paper. So this is thick paper. You get this paper specially from herbarium supply companies, and it's thick paper. It's made especially for mounting plants. We put a ventilator below so we can have a space to work on. We're going to move that over here. Some other materials that we need to mount plants. We need some weights to weight down the plant after we've applied the glue. We need some glue, and then we need some special tools here in case we need to trim on the plant at all. We're also going to add some fragment packets, and fragment packets are just these acid-free folded pieces of paper that allow us to put material in them. So now we're ready to mount our plant. So we sit down here, we've got our nice sheet of paper ready to mount. We've got our specimen open. The first thing we want to do is we want to apply the label. And to apply the label, we want to add a decent amount, but not too much glue to the back. We want to kind of spread it around. Not too much glue. And then we take just a regular little sheet of scrap paper and kind of spread that glue around a little bit, getting it near the edges here. Just making sure we don't have any big bubbles or gobs of glue. Kind of spread that glue around. And then we apply it to the bottom right-hand corner. That's standard for herbarium sheets, the bottom right-hand corner of the sheet. So I've added my label there. I'm going to put some weights down here to keep that label weighted down. Now we want to move over to our plant, and we want to kind of place it on the sheet to see how it might go. So that looks perfect right there, right? As you can see, when we press this plant, as we talked about, we have both top and bottom portions of the leaf. Everything's kind of non-overlapping. So this is a good looking specimen to mount. So we flip it over, and we start applying glue to the back. Now we're not going to glue it down everything. We just want to apply glue fairly liberally to many portions of the plant. We want to leave some of the leaves free in case someone wants to take a leaf off later, so we're not going to glue down everything. So I'm adding some glue to most portions of this. I'm going to put some glue here along the stem, so that stem will get nice and glued down. That's important. Put some glue here along this stem, along that stem. You can do this over your newspaper, so in case you have any drips, you're not dripping on your herbarium sheet. And then we can just move over and apply that to the herbarium sheet. So just lay it right down there. And then we can start adding some additional glue. So we can start putting some glue underneath places where the stem or the petioles are kind of coming up. We can squirt a little glue kind of under. We can even kind of cover these portions to kind of tack down certain portions of the stem. You can tack that down there, tack that down there. Put some glue under here. Squirt some more glue under the stem there. And then we can start adding some weights. So we'll go over here to our weight box. You can see we have weights of various sizes. Some really big ones, some medium ones, and some small ones. I'm going to add a big one here to this stem here to make sure that stem gets weighted down. Then I'm going to start adding some weights over the top of some of those places where I kind of tacked it down. You can see the glue is in the middle. These circular washers are great for this because they leave a space in the middle. All right, so I'm just adding some weights over the top of where I did that. And you can just kind of put some weights down on a variety of places that you glued down. This specimen's ready to go. It's ready. We've mounted this specimen. We've added the glue. So what we're going to do now is we're going to move it onto a stack that we've already made here. we're going to add some bottle caps to the specimen here. That way we can stack another specimen on top of it here in a few moments. Now we've mounted our first specimen. Ooh, I almost forgot. I forgot to add the fragment packet. We want to take one of these fragment packets. I'm going to add a good amount of glue to the back of that, around all the edges. Add a good weight to that. We're also going to set this came along with the specimen. These are some fruits from this plant that we'll put in that fragment packet after this plant is the glue is dry. That's what a fragment packet is for. It's to put any fruits or any small bits that go along with the specimen that can't be easily mounted, or perhaps later, as this specimen is used, a leaf might pop off, some pieces, some fragments might come off. Those can be safely tucked in there and they'll remain associated with the specimen. So we got our fragment packet on there. So we've mounted our first specimen. We can get rid of that newspaper. What's our next specimen? Looks like we've got a nice fern here. Again, you can see the locality information here. This was collected in Kansas in Geary County from Geary State Fishing Lake. I noted that it was epipetric, which means on rocks, on a calcareous ledge at the rim of a northeast facing slope above the lake. I noticed some plants that were with it. I got my location, or my geolocation there, my latitude and longitude. I noted that this thing was sporulating. It was making spores, and I even noted that it had 32 spores per sporangium, which means it's asexual. Here's my collection number and the date. And this thing actually has a specimen at Wichita, at the University of Kansas, and at the University of Minnesota Duluth. So anyone that looks at this label knows that there's duplicates at 2 other herbaria. All right, so let's get busy with mounting this guy. Get us another ventilator. Let's work on the label. Get that glue nice and spread around there. Affix the label. Go ahead and get it weighted down. Now let's start adding these. Now we have several individual plants here. We can kind of place them on here so we can get a sense of how they're going to go on the sheet. Make sure we got plenty of room. Let's go ahead and apply our fragment packet to the top. Fragment packet can really go anywhere. They usually go at the top of a specimen. Let's go ahead and get this fragment packet on there so we know that real estate is taken up, so we won't put plants over it. Put that there. Now I'm going to start working on these individual specimens. Make sure I get some glue on that rhizome. Apply some glue along these leaves. Little drops of glue there on the edges. Some of these pinnules. Drop that down there. A nice weight over the rhizome there. This is a much more kind of dainty little specimen. can use these smaller weights. Again, we want to make sure we have some up and some down. So most of these are showing the bottom side of the leaf, which is important in this plant because that's where we can see the sporangia. We'll leave at least one specimen here on the top. We'll leave it up, just so we know what the upper surface of the leaf looks like. Maybe we'll put this one up too. Looking over my newspaper here because I drip. Well, this specimen's ready to go, right? We've mounted it. We've got one little piece here. Maybe we'll set that there, so maybe that can go in the fragment packet when the fragment packet is dry. Put a couple over here. Those can go in the fragment packet. We're going to set it up here on the top of our existing stack. Put some caps on it. So we'll be ready to go with the next specimen. And there we go. We've mounted two specimens. You can recycle your newspaper. And there you go. So we mounted two different kinds of specimens there. A woody plant and an herbaceous plant. And showed you how to apply the labels and the fragment packets and the specimens themselves. So here in about a day, actually just a few hours, but certainly overnight, the glue will be dry. We can take the weights off, and then these specimens are ready to go in the herbarium.